right, everyone, thank you for being here for our third workshop. As we get started, okay, there we go. So our agenda today is we're going to do a little bit of review off of what we did last week. We're going to discuss what the methods are, as that's going to be something that's coming up for a due date in November. I'm going to remind you all about the opportunities for 3D printing in case that's something you want to incorporate in your methods. We're also going to look at the NASEP forms wizard, since that's part of the process to make sure that we have everything we need to start conducting an experiment. Also discuss the SARSEF IRB if that's something that's needed for you all, and then just have a quick summary. And this is a reminder of our community norms. If we can please be focused on a common goal, strive to understand multiple perspectives, be willing to be disturbed or change your mind, critique ideas and concepts, not people, to share the space, stay engaged, recognize when each of us needs space, accept non-closure, and apologize and work to repair damage if a norm is violated. So in the chat or unmuting, can someone please remind me what we discussed at our last workshop, if you can remember? Feel free to unmute or put it in the chat. And here's some photos to help jog your memory. What did we talk about last week? What did we talk about last week? Anybody? Go ahead, Austin.
We were talking about how to be our element and the feedback. That's the main things I remember.
I heard feedback. You said we talked about something in feedback. We did talk about feedback. I missed the first part though.
Oh yes, we were talking about the feedback for the pre-assignments.
Yes, that is true. I hope everyone was able to review and understand some of the feedback that was given. I think, again, a lot of it was great job, but also asking, how are you going to turn this into a research question? What is your hypothesis for this research question, and what your purpose statement is? So very good. So thinking about the hypothesis and things like that, how would this SMART part here, how did that relate to what we talked about last time? If you don't remember, it's okay, because we're going to talk about it again. But yes, we talked about when creating these hypothesis, research question, things like that, how you can make it a SMART goal or incorporate these aspects of SMART within it to make it extra specific, clear, and something that folks won't, like if they ask you a question like, oh, how are you going to do that? What will you use? It's pretty clear in your write-up because you incorporated the SMART goals of things, which is specific, measurable, achievable, realistic, and time-bound. We talked about the research process, and the photo that I used here was the DNA example of a research process, but there was a lot of different photos. There's going to be another photo in here in a few slides that kind of looks at it from a different perspective, but we talked about the research process and getting started on the NASEP research guide. If there's anything I forgot, you can put it in the chat, but we'll move on to the next slide. All right, so I am going to launch a Zoom poll at this time. It's going to pop up on your screen, and if you can complete the questions. 2 of the questions are more quiz-like, like just testing your memory from last week. And then the last question is the last question is asking basically, do you have any questions? So go ahead and take the time to complete these poll questions now, and I will start a timer for about 2 minutes. If for some reason they're not showing up on your screen, Rudy, did they show up on your screen? Okay, if for some reason they're not showing up, you can put them in the chat as well. If they're not showing up, the questions that are being asked are to match the following. So maybe you wouldn't be able to do that, but it's a review of what a hypothesis definition is, also the definition for research question, and also a purpose statement. The second one is the SMART in SMART goal stands for specific, measurable, achievable, relevant, and time-bound. True or false? And then the third one is, do you have any questions related to the research project that you would like answered? And that one is just open-ended. So if there's anything that you've been wondering or hasn't been made clear yet, feel free to ask. We have about 1 more minute and then we'll close out. Forty seconds. Thirty seconds. 20 seconds. Last 10. Close it out now. Great, let's see. So let's see, share the results. Is it sharing the results to you all? Yeah, great. Okay. So a hypothesis is an educated prediction. Looks like everyone got that right. Research question is a clear, concise, and open-ended question. And it's something that most times they wouldn't want something answered in like a yes or no fashion. So almost everyone got that right, very good. And then the purpose statement is a clear declaration that explains the intent of whatever that research is. So summarizing, because I can't see exactly what I put. Okay, there it goes. And then SMART goals. I know we just talked about it in the previous slide, and so everyone got it right. That's great. Shows you're paying attention. Good job. And then I'm going to click view details here, and I'm going to just look at the questions for share results to see what folks might have questions about. I'm looking at my screen. Okay, great. It's taking me to something else to sign in. So 1 moment while I find that screen. It's not showing me. The last question, I guess I have to sign into Zoom to see that one. Or I'm just having technical difficulties, but oh well, we'll take this route. All right, I do have the questions on screen now. No questions, no questions, no questions, no questions, no questions. Got it. Okay, great. Thank you for answering that question regardless, if you didn't have any questions. All right, but if you do have questions during this presentation, again, feel free to put them in the chat or interject if you need to. So I wanted to share this website. It was really cool, and I found it because, as you all know, where Amelia is from, this is the science fair that she's most likely to compete in. And they have a really great website here that has cool videos that sort of explain each step of the process. And I'm likely going to be referring to it as we continue on with this series. And the website is here. I'll add it in I'll add it in other spaces as well in Slack, and also to the research guide if you would like to refer to it. As a refresher, I think I watched the getting started, the formulating a question, formulating a hypothesis, understanding science practices. I haven't done the SciComm regulatory bodies video, but the ones that I did watch are great. And so the one we're going to be looking at today is the science fair method and materials. But to provide a little context, as you can see here, there's a little turtle in this third video slide here. And in the methods in this video, the methods and materials that we're going to be watching, it refers back to this formulating a hypothesis video here. And so I'm just going to go over it a little bit to provide some context. So within that video, the student is asking, when presented with a cricket or melon, which food will cause a turtle to move across the cage the fastest? So they have a turtle that they want to feed, and when they present the turtle with either one of these options, they want to know, is the turtle going to hurry up for a cricket, or is the turtle going to hurry up for the melon? This is the research question that this individual is asking. So as a reminder, the hypothesis is an educated prediction about the experiment. And so if this student is, I think they call them Mohammed in the video, if Mohammed is formulating a hypothesis, this would be the structure here that he could use. So if blank happens, that will cause blank to happen. So in the context of that research question with the turtle, it could be like, if a cricket is placed at the end of the cage, that will cause the turtle to go faster than if presented with a melon. I actually think I have them flipped, but the next slide will let me know. So yes, so this is the first version of the hypothesis that was created. So if crickets are presented to the turtle, then the turtle will move faster than if we presented a melon to the turtle. So this is great so far. Any notes so far with this hypothesis? Any notes? I think it's pretty clear. You can see what, like, if what variable they're deciding to use. There's a comparison, but there's room for improvement in a way if we apply that the SMART acronym to it. So the new version is a little bit more specific. So when presented with a cricket once a day for 1 week, so that added the time aspect to it. The red-eared slider, they added the type of turtle, because maybe different turtles like to eat different things. You don't know. That's why it's great to put a more specific type of turtle in this scenario. That turtle will move across its tank with a lower average time. So that's the measurable aspect of the hypothesis. Then when it's presented with cantaloupe cubes at the same rate on a subsequent week. So I'm going to go back just for that comparison. So the first version was, if crickets are presented to the turtle, then the turtle will move faster than if we presented a melon to the turtle. Now looking at this one, there's a lot more details, right? And with these details, it also gives rise to possible things that could be included within the methods, right? So when presented with a cricket once a day, so Mohammed's going to be presenting a cricket once a day for 1 week. That's included in the methods because it gives the time frame for when he's going to be doing it and how often. And it also includes average. So if Mohammed is wanting to take an average, that means he's going to have to time the turtle at specific times and divide over that time frame to make that comparison. And he also included that he's going to do one variable for 1 week. So maybe a cricket for 1 week, and then the next week do the cantaloupe cubes. All right, let's continue. They didn't include this in the video. They did include the different hypotheses in the previous videos, but they didn't necessarily include a purpose. But since we are asking you all to include a purpose as part of your assignment, I developed this purpose based off of what was learned in the videos before for context. So I wrote that the purpose of this experiment is to compare the average time it takes a red-eared slider turtle to move across its cage when presented with cantaloupe cubes or crickets to see if the turtle will move faster for cantaloupe cubes. So if you go back to the previous slides, you'll see that it's all very similar in terms of the wording. It's just how you present it might be different. So of course the research question is going to be presented in a question type way. The purpose is going to be more like, this is what we're doing and why we're doing it. Any questions so far? No? Okay. So that is the context for what we're going to be doing. But since this week we're going to be talking about methods, this is the area of the scientific method that we're talking about. So the design process, the methods for this experiment. Where is this video? Sorry, I need to find the video in the chat. Okay, so I'm gonna go ahead and share my screen for what they decided to do. Any questions so far? Make sure that I shared with audio, share sound. Okay, great. So this is the website that I had presented earlier that has all the different videos. We're going to be looking at the methods and materials. I'm going to play it and I'm going to ask if someone can make sure that they can hear it. Hopefully we don't get an ad. Oh, we did get an ad.
At Cox, we're always a step ahead. Like how the price of your plan won't go up for 3 years. Guaranteed. That's refreshing. Ooh, s'mores.
Lock the price of your plan for 3 years. Just another way we keep you a step ahead.
Were you all able to hear the ad? Okay.
Hello, I'm Dr. Erin Golden, Director of the Denver Metro Regional Science.
Sorry, I do watch videos at really fast speeds. I'm going to slow it down. We'll do normal.
Science and Engineering Fair. And today, I'll be talking to you about how to build a materials and methods list for your science fair project. After deciding on your research topic and identifying your research question, you'll need to plan out the steps you will take to test your hypothesis. This is called your experimental method or procedure. Your experimental method not only includes the steps you will take to test your hypothesis, but also the materials you will need to complete these experiments. Be as comprehensive as possible when making these lists. Think through each step of the experiment and identify everything you could possibly need for safe and accurate data collection. For example, in our previous video on formulating a hypothesis, Mohammed outlined an experiment to test which food will cause a red-eared slider turtle to move across its cage the fastest. If you haven't seen this video yet, be sure to check out the complete series on the DMRSEF YouTube channel. For his experiment, Mohammed plans to present a turtle with one of two foods, either crickets or cantaloupe, once a day for 2 weeks, and time how long it takes for the turtle to move across its cage to reach that food. First, let's think about the materials Mohammed will need to complete this experiment. The most obvious materials are those directly involved in the experiment: a red-eared slider turtle, freeze-dried crickets, cantaloupe, and a cage. Mohammed is planning to time the turtle's movement, so he'll probably need a stopwatch as well. Going a little bit further into our planning, we should think about the steps that Mohammed needs to take before beginning his experiment. For example, he will need to cut up the cantaloupe, which requires a knife and cutting board. Perhaps as an experimental control, he'd like to make sure his cantaloupe pieces are the same size for every trial, so he'll need a ruler as well. If he's planning to use tweezers to handle the crickets, those would go on his materials list too. How about gloves or hand sanitizer? From doing some background research, I know that turtles can potentially cause salmonella, so Mohammed should include these safety precautions in his planning as well. It is important to make sure you have everything you need before beginning your experiment. One way to make sure of this is to work with your adult sponsor or qualified scientist. Ask them to double-check your materials and methods list before you begin testing. Talk through each step of the process to make sure you have thought of everything you could possibly need. It's frustrating to have to stop an experiment halfway through because you forgot something obvious. Getting back to our turtle experiment, let's list out the steps in our procedure and make sure we have everything we need. For step 1, we'll need to prepare the test food. If this is a melon trial, we'll use a carving knife to prepare a 1-inch piece of cantaloupe. And if this is a cricket trial, we'll need to remove 1 freeze-dried cricket from the bag using tweezers. Next, we'll place the test food on the far end of the cage. While wearing gloves, we'll then place the turtle on the other end of the cage, approximately 8 inches away from the test food. For step 4, we'll start the stopwatch and observe how long it takes for the turtle to approach the food. Finally, we'll record observations in our lab notebook, then repeat steps 2 through 5 each day for 1 week. Do we have everything we need? Looks like we do. Awesome. Creating your materials and methods list is a great time to think ahead to your safety form. Are there any materials or procedures that could cause significant harm to you or others? Are there any procedures you'll need to have adult supervision for? All of these will need to be addressed on your risk assessment form and should be included in your project plan as well. Remember to be as specific as possible. Thanks for watching. In our next video, we'll explain how to collect your data.
Helpful resources. All right. Any questions about that video?
Visit our website.
Oops, yes, we're going. Any questions about that video so far?
I just been a meeting, but it was in medical, and I just got up. But to join now. Sorry, adjourning.
Okay, yeah, no worries. Thanks for being here. And we recorded. So if there's anything that was missed, then hopefully it's covered in the recording. Any other questions? Okay, so where is my presentation? So many screens. There it goes. Okay. All right. So to remember what we just discussed in this video, we talked about the experimental methods. And so this is the procedure that helps us test whatever hypothesis that we have. And I want to ask you all, what were some key takeaways from that video? What are some key takeaways from that video? What are some things that stood out to you? Something new you might have learned?
Can I add something?
Yes, of course.
I thought it was interesting how in depth she went on the materials list of like needing knives and gloves for like cutting the cantaloupes. Because I would just think, oh, it's like a part of making it, so I wouldn't include it on a materials list. I thought that was interesting.
Yeah, being very detail oriented, right? Somebody might have stopped and just been like, I need a turtle and I need some food, right? Maybe my phone. But yeah, she did really reflect and say, like, actually, you need to think about what happens during, what happens before. So thank you for sharing that. I'm going to check the chat. Leah said making sure that all of these safety risks are written down. Natalie said being specific about the list. Yep. And safety precautions. That did stand out to me. Because through research, we do not want to cause harm to anyone or ourselves. So safety is a big factor to think about. So thank you all for sharing. I might have missed a few things, so you can feel free to still put stuff in the chat. But things that stood out to me, at least, were thinking of every step of that experiment. So the before, during, possibly after. I was supposed to say think of every item you need at each step. Something that stood out to me was the controlling for the cantaloupe size, because someone might just chop it up, but if you want it to be a good comparison, being specific on how big you want the cantaloupe sizes. So even including the ruler, if they want to make sure that it's comparable each time. You all brought up the importance of safety and how doing some background research might be necessary, because handling turtles, someone might not know that you can get salmonella for turtles. So if you weren't taking the proper precautions to like have gloves or not washing your hands, then it could pose a risk to like yourself or other people, you know, touching different surfaces and things like that. So safety is a big thing. And so I guess take a moment now to just think of, okay, with what I'm thinking of, what might be some of the steps that I would include? What are some things that I might not have thought of yet? And what might be something that I need to look up to make sure that what I'm doing is okay? So again, take a moment to think of, depending on what it is you're wanting to do, what steps that you might need to take, any materials, what are some safety concerns, and what might be some sort of background research that you might have to do to make sure that you're all good in your experiment. Just jot it down on a piece of paper. Put it in your notes on your phone or your iPad. And we're not going to share it out. It's just an opportunity for you to self-reflect. So finish up whatever it is you might be thinking about or writing. But thank you for taking that moment for reflection. You can continue to write, as some of these things here might be helpful as I review it. So again, list methods used, list materials used, I mean. Consider how many samples you might need or how many trials you might need. So I wouldn't necessarily say that there was samples in the samples that were in the previous example, but it could be that the materials that are needed, since he's doing it for, Muhammad's doing that experiment for a week, that he would need at least 7 crickets. Maybe he would add one more or something in case it got lost, but he would have at minimum 7 crickets to feed, and also 7 slices of those cantaloupe, right? In the instance where if somebody was doing something with like water, because we do have the water test kits, if that's something that you want to use for your experiment, how many samples would I want to collect if I'm deciding to do a comparison between one site and another site? Do I only need 1 sample from that site and another sample from this site, or do I want to do it twice to make sure that my results are consistent throughout? I don't know if in that video, I don't recall if Muhammad wrote down like a specific time for when he wanted to do this. If he's like, I'm going to do it at 12 o'clock every single day to make sure that it's consistent. That might be something that you would consider if you're doing something that's temporal or overtime, to say like it has to be done at this specific time for comparison purposes or just to make it consistent. Because things could change between like the morning or the evening, depending on what it is that you're asking, you know. If the turtle, for instance, is more hungry at a certain time, it might be best to just keep it at the same time versus having it at different times. If you're surveying people or asking their opinion, maybe not doing it at a time when they would be tired, you know, so figuring out when you would want to provide that survey or whatever. Thinking of the location that you would have to go to, going to the 4th bullet here. So the source of your samples. I know that there was a student that wanted to make comparisons between like water sources between like Old Pascua or New Pascua. So those are the sources of your samples. If you're wanting to do something that's like survey based, where do you plan to get your like study population from? Is it at your school? Is it at a community center? And also to make sure that you're writing down your observations for what it is that you see. If you're doing soil or water collection, what does it look like on that day? Is it rainy? Is it sunny? Is there, for instance, if you were going up to like the edge of a lake or something, like what are things that you observe around it? Is there like moss? Are there animals grazing? I think is it by something that could cause leaching of any sort? So note down like what you see, the time. Those are all very important to include and to write down, because I know a lot of the time we think we're going to remember, but that's not always the case in terms of memory. I like to take a lot of pictures because it has like a timestamp. It has like the location and things. So that helps me remember. And also, what are your test variables? What's the independent variable? What's the dependent variable? Are you making a comparison? And what would be the control if you're making that comparison? The independent variable is the thing that you're usually controlling, and then the dependent variable is the thing that you're not necessarily controlling. So, for instance, in the turtle example, the independent variables are the food items. So the cricket and the melon are independent of each other. The dependent variable in that situation is the time that the turtle takes to go to the end of the cage. So in that situation, the food items are the independent variables, the time is the dependent variable. Any questions so far? Okay. And again, if you need to take a second to write down things that you think you would need for your experiment, feel free to do that. I know that sometimes when you're hearing it and seeing it, ideas start coming up, so feel free to do that along the way. And observing things often takes practice, and so observing is a very natural thing to do, but you want to make sure that you have things that you're paying attention to in terms of the timing of things and whatnot. I don't know if you all have heard of the windshield survey, but what they were noticing is that there was a lot less bugs on windshields, and people noticed that, and they were wondering what the cause of it was. So that was a research question that people were just observing and thought of different ideas, but they actually decided to test what the actual reasoning was. And so it ended up being linked to the decline in bugs. So it's really interesting because that's something that people just noticed and was like, I'm noticing that there are fewer bugs on the windshield when we drive. And someone actually decided to test it and see what the cause of it was. And so having those very detailed methods is important because you could have a research question, but figuring out how to test it is also a very important part of research. And again, that second bullet is just a reminder that memory is often selective and unreliable, so having those notes is important for your lab notebook. All right, in the chat, may someone, or feel free to unmute, can someone remind me the difference between qualitative research and quantitative research? Qualitative research and quantitative research. We got qualitative and quantitative. Any ideas?
Isn't quantitative like number based?
Mm-hmm. Yeah. So if quantity is numbers based, what would qualitative be?
It would be the qualities or like almost like physical properties maybe, or like stuff like that. Just more physical like observations rather than numbers relating to the quality.
Great, thank you. Yeah, so thank you for providing those responses. You all hit it on the head. And so within those different like paradigms, there's also different tools that could be used to test something that's qualitative versus quantitative, right? And so, as was stated, the quality of things, you're looking at maybe like themes or the look of things or changes that aren't based off of number. So if you're wanting to do something that's more qualitative, you might interview people about like perceptions or their experiences. You could have a focus group. I guess focus group in this sense would be different because interviews would be maybe more one-on-one, where a focus group is in a like in a group setting. You can make observations about something. So looking to see how things change over time, or after something happens. A document analysis might be looking at something like a book or papers and seeing what the themes are or what the commonality things are within there. And that's usually maybe secondary data. It's not something that you're doing yourself, you know, like you could get papers from a place and from there look at it. It could be like hospital notes or satisfaction scores, and not scores because that would be quantitative, but yeah. And oral histories or life stories. And you all who did the first option for the pre-assignment did some interviews. So you already did some things that were qualitative. So good job. And I guess the oral histories or life stories would be different than the interviews because that's more focused on someone's like personal life experiences, where interviews you might be asking them like a variety of questions that are not based off of their personal history. So let's move along to quantitative techniques. If you're wanting to do something that's more quantitative, you might be incorporating surveys or questionnaires in your work. You could be making observations. I know that observations is seen on both sides, but observations in this sense could be if you were coding what your findings were to specific numbers. So if I was taking something that was like on a scale and I'm like, this is going to be 1, a 2, a 3, because you're still quantifying those things in a way. You're not necessarily looking at the theme. Again, document screening. So this could be if you were counting like word occurrences or the number of times that something happened. And then experiments. That's the main one that most know. And then even within experiments, there's a lot of different types of experiments as well. And I also want to point out that something that's not talked about as often, I think it's mainly for simplicity's sake, is there is another type of research that actually incorporates both qualitative and quantitative techniques. It's called mixed methods, and it's how it sounds. It's if you add both types and are incorporating it into your research. So for instance, you might give a survey, but you also might be doing some sort of interviews to dig deeper into some of those questions that aren't captured in the survey. Any questions so far? No? Here are some examples of experiment types. I'm saying some because again, there's a lot of different types. And so for the context of like geoscience or water, here's some photos here that represent natural, controlled, and field research. So natural is more observational in a sense because you're not making any sort of change to the space or the variables. There could be things happening, but you're specifically not making that change. An example could be looking at coral bleaching over time. You know what I mean? You're not specifically going there and bleaching the coral, but you're looking to see the changes that are happening as a result of the populace, maybe because people are using sunscreen or different things. It depends on your research question, but you're not specifically making those changes. You're observing. A controlled experiment could be something like in a lab. A great example is the biosphere. Remember when we went to the landscape, I don't remember what the E and the O stand for, but those huge beds that they had that had all that soil and different things in there, they were able to control the moisture levels, anything that they sort of introduced. So that was a very like controlled environment. And in this photo you see here, it's like you're watering one thing of flowers, but you're not watering the other one. So like what happens, you know? So that's sort of like a more controlled environment space. And field is you're going out into like the community, not the community, but you're going into the field, and you're just sort of seeing what nature is doing, but you're still applying that water to something and still having like a hand in that experiment versus natural, because sometimes natural and field look a little bit similar, but the field research is one where you're actually applying some sort of change to like a variable. Any questions so far? Depending on what your research question is, what you're asking, it will depend, like, it will depend on what sort of experiment you might decide to use. So for the folks who are collecting like water samples and doing a comparison, I wouldn't say that it was like a controlled sort of space or things like that. But if you were taking water sources and deciding to put something in there to see what changes, then that could be considered like controlled or not controlled. This timeline here is just another example of like a temporal type of study. Are there graphics in here? Yep, there's graphics. So the temporal studies usually want to test if there's like a relationship between two variables. You would set a time frame. So if you want to look at it over 2 weeks, days, or months or something, and begin this experiment with sort of baseline data and then seeing how something changes over time. If you're doing these ribbons here sort of designate like if you have a specific spot that you're going to and want to make sure that you're going to that sort of spot. And so for instance, we get our baseline data here, we mark our spot, and then a huge like wind event happens, some sort of weather event happens. And then after some time passes, I record that this event happened, and then I notice that there's this new type of lizard or animal that's in the area. Those are things that I would write down and observe. I could also include that, like the number. I could also write down things that possibly could have resulted in this change in animal being like introduced, you know? So that's sort of an example of a temporal thing. You're looking at something over time and recording those sort of observations. This one is an example of a comparison, but I'm not going to really go over this one because I would want to talk to you all about the opportunity for 3D printing. So any questions so far about experiment types, different tools that you can use, if it's qualitative versus quantitative? So I want to remind you all that we have people here at the university, like I'm sure you all remember Nico from the BattleBots, who is heavily involved with the Catalyst Lab. And I think some of you might have done some 3D printing while you were here working with Nico. But this is helpful because if you're thinking of designing something, you're like, I want to use something that looks like this, but I can't really find it. It's not something that would be easily findable at like a store or something. You can 3D print it and see if this prototype is useful for whatever experiment that you're doing. It can be inexpensive. It just depends on what is involved with your methods. Like if you're using some sort of chemical, maybe that wouldn't be best to use something that's 3D printed as it might degrade. But it's a great opportunity to use 3D printing if it's if it's something that you're looking for in terms of like design or filtering something or prototyping something. So consider that in your methods if it's something that you want to 3D print. I know Nico is very open to providing assistance. And if you need help figuring out how to do some of these things, we did 3D printing in more heavily in previous years. And so there's someone named Dr. Budanov who does things with material engineering. She'd also be able to provide assistance as well. So yeah, a lot of different 3D printing resources. This is Dr. Budanov's email. If you do want to use 3D printing, please let us know, and we can sort of figure out a time frame. If you're needing to like prototype something, we can get it printed, send it to you, and then if you need to make changes, print it and send it to you. So just factor that into your planning as you're developing your methods. Or these are the other resources that could be possible in the area that you live. Your school might have a 3D printer too, so different things to consider. All right, so in discussing the methods, going back to that theme of safety and protocol, making sure that things are being followed correctly, the Society for Science, who hosts the International Science and Engineering Fair, has this website here that I'm going to take us through, since this is part of the assignments for the coming week. And these are the steps. Okay, so for the purpose of this activity, we're going to go back to Mohammed's turtle hypothesis and act like that's the question that we're asking. And so we're going to go through these steps and answer it based off of that. Okay, so basically says welcome, move through this wizard, previous or next buttons depending on what we want to do. And so the first question is, will your experiment include human participants, vertebrate animals, recombinant DNA, microorganisms, human or animal tissue, hazardous chemicals, activities, or devices, or none of the above? So did his experiment include human participants? No, right? So we're going to skip that. However, if yours has human participants, you would click this, of course. Did it include vertebrate animals? So that means like an animal with spines and bones. It's not like a microorganism like an amoeba or something like that. It did, because there was a turtle involved, right? So we get a warning here. It says, note, there are limitations on the types of vertebrate animal projects that can be conducted. It is strongly advised that you review the vertebrate animal section of the international rules. Vertebrate animals, as covered by these rules, are defined as all non-human vertebrates, including fish at hatching or birth, live non-human vertebrate mammalian embryos or fetuses, tadpoles, birds, and reptile eggs starting 3 days, 72 hours prior to hatching, zebrafish past 7 days post-fertilization due to delayed cognitive neural development, and cephalopods are to be treated as vertebrate animals. So note, a project is not considered a vertebrate animal study if tissue is obtained from an animal that was euthanized for a purpose other than a student's project. So very detailed. And there would likely be other pop-ups as we go through this, but those are important to read because there could be questions about like what vertebrate animals are, and that definition was very clear in that pop-up. So from that definition, yep, that would include turtles. So will your experiment be conducted in one of the following sites? It didn't specifically say, but I'm assuming that he's going to do that at his home. So please note there are limitations to what types of projects can be done in a home environment. Projects involving potentially hazardous biological agents must not be conducted in a home environment, and there are more strict guidelines for vertebrate animal projects conducted in a school, home, and field site. It is strongly advised that you review the appropriate sections of the international rules. All right, noted. Next question here, there's two in this page. Is your project based on prior research that you have done? We don't know, but for the purpose of this, I'm going to say no. This is a brand new experiment. All right, next page. It's based off of human participants. Will your proposed experiment involve humans by including any of the following? Surveys, professional tests or questionnaires, emotional stress, for example, asking questions of a personal nature, exercise or physical exertion, ingestion or tasting of any substance, student designed invention, app or prototype, none of the above. So for the purpose, he's observing and tracking the turtle based off of time. So it doesn't include surveys, questionnaires, and things like that, none. And I don't think it includes any sort of human participants. So it's likely going to be none of the above for all of these, but we're still going to read them to make sure that everything's clear. Will your experiment include interacting with any of the following groups? Children, pregnant women, people with disabilities, mentally disabled persons, economically or educationally disadvantaged persons, persons who suffer from a medical condition or disability, adults over 18. Nope, none of the above. And the next question is, my project involves a review of pre-existing human data sets or human data obtained through record review. Nope, does not. If yes, how did you receive the data? We do not have to answer this because it doesn't include it, but just for the purpose of this. If yes, these are the options. It was not anonymous or de-identified. It was de-identified and anonymous. It was publicly available data. All right, next page. Vertebrate animals. Probably have to reply something with this because we do have that in our experiment. Your answers address your possible use of vertebrates animals, and it will help determine whether the project can be conducted at school, home, field, or must be conducted in a regulated site with a proper IACUC approval. Will your proposed experiment include any of the following? Study of vertebrate animals in their natural environment or zoo? I'm not sure. Study of livestock using standard agricultural practices? Nope. Study of fish using standard agricultural practices? Nope. Study of fish from the wild? Nope. Study tissue is obtained from an animal sacrificed for your project? Nope. Tissue is obtained from an animal sacrificed for a purpose other than your project? Nope. All other studies with vertebrate animals. Okay. So when I clicked all other studies with vertebrate animals, it says, please review the completed vertebrate animal section of the rules. All other studies will need to be conducted in a regulated research institution with proper IACUC approval. The following types of studies on vertebrate animals are prohibited: toxicity studies, any study involving adverse stimuli, studies of pain, learned helplessness studies, predator/prey experiments, and any project that causes more than momentary pain. But I don't think I would include none of the above here because none of the above would suggest that we don't include any vertebrate animals when we do. So next page. This is related to potentially hazardous agents, biological agents. Potentially hazardous agents, biological agents. That's a mouthful to say. Will your experiment include, check all that apply, bacteria, viruses, viroids, rickettsia, non-edible fungi, cyanobacteria, or parasites? No. And I know that there is a chance of contamination from salmonella because of the vertebrate animal that we're using, but that's not specifically what we're studying. So that's why I wouldn't check bacteria here, because that's not part of the research question. That would go under like our risk assessment. So BSL-1 organisms, BSL-2 organisms, multi-drug resistant organisms, working with E. coli, OP50, and other strains of E. coli that are used solely as a food source for C. and glands, prions or prion-like proteins, none of the above. We're going to click that. Next page. Tissues and bodily fluids. Will your proposed experiment include any of the following? Research involving human and or non-human primate established cell lines and tissue culture collections? Nope. Culturing of samples from fresh frozen tissues or body fluids or meat? Nope. Collection and examination of fresh frozen tissue or body fluid meat products? Nope. Human breast milk? Nope. Human or wild animal blood or blood products? Nope. Does not involve any of that. Hazardous chemicals, activities, or devices. Will your experiment include these hazardous chemicals? This includes household chemicals, activities, or devices. DEA controlled substances, prescription drugs, consumable alcohol or tobacco, firearms or explosive, radiation or radioactive substances, chemicals, regulated drones, hazardous devices. None of the above. All right, so this is our summary here. We're at the very last page. And so the answers you gave to questions on the previous pages of the rules wizards indicate that you will need to download the following forms in order to adhere to the international rules. These 3 forms are required for all participants. So even if you don't complete that form, this is what you would have to for sure do. The form number 1, which is the checklist for an adult sponsor. The second one is student checklist and research plan. And the 1B form, which we collected from you all in the summer, actually. So this one we already have on file, the approval form. We also need the official abstract form. This one here, this is for every form should have, or every science fair has an abstract that they usually ask for. Some use this official abstract form, but when we had a student who participated that was coming from Massachusetts, they had a different form for their science fair. So in most cases, you might use this, but it might be dependent on the science fair that you enter in. Just as a note here, because we do have an animal in our experiment, we would have to fill out this form. And there's also a second form here, the B form. The difference here is this one is for a non-regulated research site. So I'm guessing that's because we said that it was happening at home. And if we were doing it in a lab of some sort or at a school, this would be the one we would have to do B, because it would be in a regulated research institution. But for Mohammed's experiment, I think we would do 5A instead. So hopefully this helped you see the steps and sort of the process that you would go through when completing this. The results would be different depending on if we had human subjects, for instance. You'll likely have to have the human subjects form filled out. If you're doing something that's survey related, they often, well, for any sort of research that's involving humans, they will ask for IRB approval. And when you go through the steps of the IRB approval, they do ask for things like if you're doing a survey, they want to see what questions that you're going to be asking to make sure that they're appropriate, that they're not going to cause like undue stress to somebody. So you'll have to include that. What else? I don't think anyone's going to be doing anything with like bacteria or any sort of chemicals and things like that, but that also has different forms and whatnot. And so as you develop your methods, it's going to be important for you to think through some of these things and have sort of a plan, because if you were filling this wizard out without having that plan, then what it gives you back at the end might not be accurate. So that's my advice for that. I'm going to switch through pages again, but we can take a moment to see if anyone has any questions on what was covered so far. No? If you're doing something that's just with water samples, that generally doesn't require IRB approval. You would still have to consider certain risks. If you should like wear gloves to make sure that you're not exposing yourself to any sort of contaminants within the water. But yeah, because it doesn't include human subjects, you wouldn't necessarily need that. I realized I didn't spotlight myself. All right, so we went through the ISEF wizard. The SARSEF organization has an internal review that they can do for students. And so what's involved with and who is involved with in a review, they generally say that you need someone who has like a psychology background, someone in the medical field, and someone who's like an educator. So say, for instance, if you wanted to create your own IRB group, your own internal review group, those are the things that those are the type of people and the backgrounds that you would need. Someone who's in the medical or health field, someone in psychology, and an educator. So if you were thinking of a school setting, you could say, okay, I'm going to ask my school nurse because they understand the health background aspect of things. I could ask the school counselor because they could be prepared in psychology, and then you could ask a teacher because they're in education, or you could ask your principal, but someone who is a teacher there could work. However, if you didn't want to go through the steps of finding those people, having them review everything, SARSEF does have a review board already. So that's really great because it kind of can save time. However, there are multiple people who are submitting to ISEF. So when thinking about your methods and what it is that you want to do, remember the time frame it takes to get approved. I believe that SARSEF said that they would review things within 10 days, or after 10 days at least, to be able to give you a response back on whether something's approved or not approved. And on this website, which is included in that NASEP research guide, they say the things that you need in order to submit. So when we did this for a student, the student wanted to do something based off of language. They wanted to see if like a traditional way of teaching language versus something that was gamified, which one would have better success. And so she wrote up her methods, had to include the procedures for what was going to be asked to the individuals because there was like a survey. And then I think they also asked like for an informed consent form because it did include human subjects. to basically say like, I consent to participating in this survey or this learning process. And then the other item that was needed was like the risks, you know. There wasn't anything too risky, but just for the sake of the activity and having something to submit, we included like maybe someone could get frustrated because they weren't getting the answers, or just the process of learning something new could be stressful, you know. So those are things that we included in that write-up for of like for the risks. And so we sent that in. They got approval from SARSEF and were able to proceed with their experiment. So these are some upcoming deadlines for our project. The previous deadlines that happened were for the hypothesis, research question, and purpose. So if you're still working on that, continue working on it, because that's going to help you with the next step, which is the materials, methods, and procedure, and also that ISEF form and IRB check, which we just went through. So that will be due on November 3rd, so in about a week. If you need more time, feel free to let us know. Hopefully by then, we'll be able to pretty quickly provide feedback and advice on what you've submitted, so that by November 10th, you can start your experiment conduction. This is going to be different for folks who maybe need IRB approval, because you can't start your experiment until it gets IRB approved, if that's what you need. So you might start a little bit after this November 10th date, but this is a good time to sort of give you some give you a good starting point to be able to do what you need to do before December 10th. It gives you about a month, because that's when we want the results to be submitted to us. And so between that November 10th and December 8th timeframe, we do have 2 workshops where we'll cover the topics of writing up your results. So if you want to use graphs or certain things like that. And then December 9th, we also have another workshop. This will be the last workshop for the fall/winter. No, fall actually. And then December 22nd is when we would want you to write your conclusions, abstract, and have your presentation draft. We'll give you feedback, and then by January 5th, do that second round of edits, and then by the 26th of January, have that final project done, so that we can start preparing for the oral presentation component of your presentation. And I had some questions from folks about what's next after the pre-assignment. So if you have those questions, please review the NASEP Research Project Guide. And again, just to review, you have the option of creating a Google Slides, a Microsoft PowerPoint, or iCloud Keynote presentation that you're going to be sharing with us. And within that shared presentation is where you'll be updating your hypothesis, purpose, methods, all those things. And we can just go in and check it, because you already shared that that presentation with us. So as an example, this is one from your peers. So they chose to do the Google Slides route. So this is instruction. Share it with me and Sophia. First step, start figuring out what science fair or showcase that you want to compete in. They stated which one and what deadlines that they have to meet. And this is important for us because Sophia and I are going to be helping you all register for those science fairs. So we need to know when we need to have those forms and things completed for you. So For this person, we actually might have to speed up the timeline that we have, because I think that January 28th or something is when I said the deadline is for the final project. But since their registration deadline is before that deadline, they're going to be sped up a little bit. So here on this slide is where you would put your research question. And so this is their research topic and the research question. I guess feedback that I would give here is it's not necessarily formed in a question, but the kudos to this is that they have the topic and sort of what it is that they're thinking about. So the content is there, which is great. That's the important thing. So the hypothesis here is if different materials are exposed to the same temperature source for the same duration, the denser ones will retain and transfer heat at a greater rate. The materials that are too porous will not be able to retain and transfer heat at a significant rate. Great, that's a good first draft. So the content is there, which is the important part. And then the purpose statement. So the purpose is clearly stated here. So to determine the differences between earth materials and how they would transfer energy and why it's important. So this is also something that is great, because it's like, well, why are you doing it? What would be important about that? So those were the items that were due. And these are the items that are coming up. So since we just talked about our procedure, here's where you would include your materials. Here's where you would include the different steps for your process. This is the website in which we looked at the forms that are needed. Here's where we ask you, what forms do you need? Here we ask, do you need informed consent? You would let us know here, and then we would be able to check like, okay, this person is using whatever, and we have to go through some sort of process to make sure that they get approval. So that would notify us here. Yeah. And it goes through a lot of different things. It has even the final draft reminder here. So that's the next steps. Let me switch screens. So we talked about that guide. So we're closing out. What we did today was we reviewed our research question, hypothesis, and purpose statements. We discussed what's involved with creating your methods, reminded everyone that 3D printing is an option if that's something you want to include in your methods. We also looked at the ISEF forms wizard and went through the step-by-step process, and also included the IRB approval process and what would be needed for that. As a reminder, for those interested in applying to the University of Arizona, the early action deadline is November 1st. And so you get priority consideration for admissions, merit aid, and also if you're interested in the Franke Honors College, then please get your items together. If you have any questions, please reach out to us. We're here to help. And we also have our contact who presented last week, Marwan Clark, who is also happy to ask questions. In the links in our emails and on our YouTube channel, there is the recording from that session. If you happen to miss it and wanted to view it before applying, it's there. Our next workshop is going to be in 2 weeks and is going to be on November 11th at the same time at 5:30. I believe by this time, daylight savings will have occurred. So for those of you who are in areas that observe daylight savings, it'll be happening at the same time as the Tucson time if you're in New Mexico. If you're in other areas, it would be going an hour back, because I don't know if you knew this, but parts of Arizona don't follow daylight savings time, including like Tucson and Phoenix and things like that. we stay at the same time while everyone else changes. And this is a quote from Wilma Mankiller. The secret of our success is that we never give up. Any questions? Any questions? Let me check the chat. So there was a question about methods for water experiments. Would I need a consent form or anything if you're taking water samples from inside people's houses? It couldn't hurt, maybe just something saying that if it depends on if it's like your own house or if it's a family member's house, then that should be fine. But if it's like you're doing like a random sample, you know, like if I was in my neighborhood and I was like knocking on people's doors who I don't necessarily know, and I was saying, hey, is it okay if I take a sample of your water? Then I would say yes, you would need like a consent form to say that people are saying, yes, it's okay that you take water from my home. Does that answer your question? All right. Great. All right. Well, if there are no other questions, then feel free to hop off. Thank you all for coming. We'll be staying on if anyone has any questions that they want to stay on and ask. Have a good evening.
Hey, Carol, thank you.
Yeah, of course.
Oh.
Have a good evening.
See you later.
See you guys later.
Aiden, did you want to ask your question? Oh, let me stop the recording.